Hello everyone, good morning and welcome to today's presentation. In this video, we are going to have a look at how a tiny spot of blood on a man's sandal led to successful prosecution, which landed him in jail. So, this will be our presentation outline. We'll have a look at what forensic science is. We will also look at the brief facts of this case. We will look at the investigative tool that the team used and why they use that tool. And we will also look at the exhibits used for the investigation and also the kinds of analysis that were performed. We will look at the laboratory process, the results they had, and the conclusion. Forensic science. So what is forensic science? The application of science to the legal arena is fundamentally one of reconstruction, that is trying to assist in determining what happened, where it happened, when it happened, and who was involved. It is not concerned with and cannot determine why something happened. That's the motivation. So forensic science cannot determine why something happened. When science is applied in this way, the adjective forensic is added, which means that it is applicable to a court of law. Forensic analysis is performed on evidence to assist the court in establishing physical facts so that criminal or civil disputes can be resolved. In forensic science, the laws of natural science are considered in making a determination about the state of a piece of physical evidence at the time of collection. Science used in court does not establish guilt or innocence, which are properly the province of the judge and jury. So forensic science cannot determine whether someone is guilty or innocent. That is the job of the judge and jury. Rather, forensic science contributes information about what may have happened and who may have been involved. It does not assert whether the action was legal or illegal. So let's have a look at the brief facts of the case, what really happened per police investigation. But before we continue, please don't forget to subscribe to this channel, like this video, comment, leave all your questions in the comment section, and also share this video. So please don't forget to subscribe to this channel. Thank you. So brief facts of the case. On 23rd June 2019 at about 6:20 p.m., a 57-year-old woman who is now deceased was attacked and killed by an unknown person on her way from her farm. Police investigation led to the arrest of a 43-year-old man who is also a farmer, and a search conducted in his room led to the retrieval of his pair of sandals, which he admitted to be his. And now, police found a tiny spot of stain on the right sandal, suspected to be human blood. Also, during autopsy, blood sample was drawn from the deceased and handed over to the investigative team for comparative analysis. Now, Now, suspect was arraigned before court and was remanded into prison custody, awaiting the outcome or results of police investigation. Now, what investigative tool did the police use and why did they use it? So they used DNA analysis. And why? You see, DNA is unique for all individuals except identical twins. What it means is that except identical or monozygotic twins, each and every individual has a unique DNA profile. So if they are able to extract and profile DNA from the tiny spot of suspected blood, then they can also extract and profile DNA from the blood sample drawn from the deceased at autopsy. Then they can compare these two profiles, whether they match. That is why they went in for DNA analysis. Also, DNA can be extracted from few cells, and same compared to a reference sample. So with modern technologies like PCR, it is possible to extract DNA from small amounts of sample or few cells. So looking at the tiny nature of the spot of suspected blood, they thought it wise to use DNA. So exhibit for investigation. What exhibits were the investigative team able to retrieve? One, a pair of sandals retrieved from the room of suspect with tiny stains suspected to be human blood, and also blood sample that was drawn from the deceased woman at autopsy. So this is the left sandal that was retrieved from the suspect's room. And you can see this area. This place has been marked. So there are some spots here that the investigative team suspected to be human blood. This is also the right sandal. Oh, sorry, this is still the left sandal. Now this is the right sandal. Also contain some stains suspected to be human blood. So some cuts from this place of the right sandal was used for DNA profiling, as well as cuts from the left sandals also. So what analysis were performed? One, analysis was performed to determine whether the tiny stains on the sandals are human blood. So the tiny stains on the left sandals as well as the right sandal. Analysis was performed to ascertain whether they were indeed human blood. Also, analysis was performed to determine whether DNA profile generated from the tiny spots of blood on the sandals, that the left and right sandals, matches with DNA profile generated from blood sample of deceased woman. So now the exhibits entered the laboratory. So how did the laboratory work start? Or how did the scientists go about with the laboratory work? So the first thing they did was to do something called serology, that is the analysis of body fluids. Then they did DNA extraction, DNA quantification, DNA amplification, that's the PCR, and also electrophoresis. So serology. They use something called a hemostasis test. Hemostasis test determines the presence of blood. It is not confirmatory for human blood, so it detects any blood at all. So after that, they went ahead and used what we call hexagon OBTI test. This kit is specific for human blood. So the first one, which is hemostasis, will tell an analyst whether what you have is blood or not. Then the second test, hexagon OBTI test, will tell you whether you have human blood or other type of blood, like maybe an animal blood. So this is the hemostasis strips. They are strips and found in this container. And this is also the hexagon OBTI test strip. No, this is a cassette, test cassette, found in this box. So this is the hemostasis test. So this is the positive and negative control from the hemostasis test. So the positive control is a known blood sample and it tested positive. The negative control was used at a place where there was no blood. So we have a negative results here. This is the original color of the strip. So it didn't turn to this dark bluish color. But once you moisten this tape and it comes into contact with blood sample, it turns to this color. So they had the same results for the tiny spot on the right sandal. Then they went ahead to do the confirmatory test for blood using the hexagon BTI test. Once again, before they used the hexagon BTI, they did the controls. So this is the negative control. This is a reagent blank. They use a reagent blank for the negative control. Then this is the positive control, where they use a known human blood. So you can see the test line, the control line all showing, meaning the sample that we use was indeed human blood and the kit worked to perfection. Once again, the reagent blank, you can see only one line at the control section, no line at the T or test reagent, meaning this test kit is working to perfection. So after making sure this test kit is working to perfection, they went ahead and applied the crime scene sample. So this is the result for the right sandal. You can see two lines, test line appearing, control line appearing. So this is a positive result. It means the stain found on the right sandal was indeed human blood. But with the left sandal, the stains were not human blood because we have no line at the test region. We have only one line at the control region. It means the kit is working well, but then the sample isn't human blood. So after the serology, they went ahead to do the DNA extraction. That is taking DNA out of the cells, that from the blood that they had. And they used this kit called the Profiler Forensic DNA Extraction Kit, manufactured by the company Applied Biosystems. So this is the kit. This is the content of that kit. So this is the laboratory where the analysis was done. That's the DNA extraction. This is where they extracted the DNA. And these are the extracted DNA samples. So they went ahead to quantify the amount of DNA that they had. That's to know the amount of DNA that they had. So they use the 7500 real-time PCR machine, as you can see over here. This is the machine connected to the computer with a software. So this machine is manufactured by Applied Biosystems. You can see the name here. And they use this kit, the quantifier trail kit. So with this kit, you can determine whether you have inhibitors in your extracted sample. You can determine whether your sample is coming from both male and female sources. You can determine whether your sample is degraded or not. So this is the kit that they use on this machine. So this is the quantifier trail kit, the pipette in the field where the sample preparation was done. After that, they went ahead and amplified the target sequences using the global filer kit. The global filer kit has 24 loci or target sequences. So that's the kit that the investigators or the lab analysts used. And they used the 9700 gene amp PCR machine manufactured by Applied Biosystems. So after amplification, they went ahead and did the electrophoresis. Once again, they used the 3500 real-time PCR machine. They used the 3500 genetic analyzer manufactured by Applied Biosystems. So this is the 3500 genetic analyzer connected to the computer with a software. So after electrophoresis, they now wrote down their results. That's the results to be taken to court. So now, court results did they obtain from the whole analysis that they did. Now, they found out that the stain on the left sandal tested negative for human blood. Also, the stain on the right sandal tested positive for human blood. The stain on the left sandal after DNA profiling gave a null DNA profile. It means an empty DNA profile. Stain on the right sandal gave a full female DNA profile. It means the blood on the right sandal was coming from a female source. Also, blood sample of deceased woman gave a full female DNA profile. So this is the table of results. These are the various loci that was tested. And this is the profile of the blood stain on the right sandal, that the sandal of the suspect. This is the profile of the deceased woman. And these are what we call the likelihood ratio. So from here, if you look at these two profiles, you can see that they are matching. 15 here, 15 there, 15, 17, 15, 17, 9, 9, 8, 12, 8, 12, 8, 12, 8, 12, 15, 16, 15, 16, 29, 30, 29, 30. It means the blood stain found on the right sandal of the suspect matches with the blood sample taken from the deceased woman. So the analyst calculated the likelihood ratio, that the likelihood of the blood stain on the right sandal coming from the deceased woman. So this is the combined likelihood ratio, 3.0797 times 10 exponent 24, a very huge number. So conclusions. What conclusions were drawn from the entire analysis? 1. The stain on the left sandal does not contain detectable hemoglobin. The hexagon obitiatis detects hemoglobin in the human blood. So if something happens and the hemoglobin level falls below the limit of the kit, then you get a negative result, even though it might be blood. So normally when you have a negative result, we don't normally say that the sample is in blood, but we say it does not contain detectable human hemoglobin. because it might be blood, but then the hemoglobin will be below the detection limit of the kids. But then when it is positive, that one we make a bold statement that it is human blood. So also stain on the right sandal is human blood. Also DNA profile obtained from blood stain on the right sandal of suspect is consistent or matches with DNA profile from blood sample of the deceased woman. Now, probability that the blood stain from the right sandal originated from deceased woman is 3.0797 times 10 exponent 24, that's the likelihood ratio, times more likely than if it originated from an unknown, unrelated individual. They use the Black American race allelic frequency. Now, in terms of percentage, this translates into a percentage probability of 99.9999%. So, in terms of percentage, the probability that the blood stain on the right sandal of the accused person coming from the deceased woman is 99.9999%. So that's quite a huge probability. So this person was sent to court again, and based on these results, he was convicted and sentenced to a prison term. So this is how science, and also this is how a tiny spot of blood on the sandal of a farmer. He never knew such tiny spot of blood existed on his sandal. After committed the act, he went home. So he never knew such an evidence existed on his sandal. But then this is the power of DNA. Through investigation and through science, he was able to be prosecuted, and then justice was gotten for this poor woman. So once again, thank you for your audience, and don't forget, as I said earlier on, to subscribe to this channel for more educative videos. Like this video, share, and also watch other videos from BB Science and Medical Media. Thank you. Have a wonderful day. Bye.